this. So new ways to create plans and the challenges of their IP protection. I will, as usual, introduce the services of the European IP Helpdesk and take a couple of minutes for the introduction of the services that we offer and what we do. So the European IP Helpdesk is a service initiative of the European Commission that has the objective of addressing current and potential beneficiaries of EU-funded projects, researchers, and especially European Union SMEs in order to help them understand what the European intellectual property rights are and how to use them. Actually, the services are not aimed exclusively to those categories of researchers or European Union SMEs, but they are open also to research and to all individuals or institutions that would like to know more about intellectual property and intellectual property rights. In principle, we are a free of charge first line support that has the objective, as I said, to support whoever is in need with regard to intellectual property questions and issues. And we do it following the services that you can see on the right-hand side of the screen. So there's trainings, website, a helpline dedicated to the questions that you might want to ask. There are also publications available, downloadable online, templates that you can use and modify. And there's a ambassador scheme that actually has ambassadors and experts of intellectual property in each and every country of the European Union, and we are also present to awareness raising events. One new thing regarding the 2021 is that we have a new website, which is ec.europa.eu/IP-helpdesk. This new website has been created in order to group together all the help desks around the world with different geographical areas of action. So we, excuse me, are grouped together with our siblings, the China IP SME Helpdesk, Southeast Asia IP SME Helpdesk, Latin America IP SME Helpdesk, and the newborn, which is the India IP SME Helpdesk. Just like them, we offer a large variety of services, which some of them I told you already, website, trainings, publications, but there's also available audiovisual content on YouTube, and on social media we are active on LinkedIn and Twitter. I'd like to focus the attention quickly on the helpline, because the helpline is probably the most interesting and important service that we can offer, because the experts that are currently sitting in Alicante are there to answer directly your specific questions, and they offer a free of charge first line IP support that, because is answering to the question that you're asking, is personal and to the point. It's also quick as possible actually. The answer to your question is going to be sent back within 3 working days, and you may ask through email, phone, and website. If you go on our website, which I told you before, you will be able to leave a query for the helpline. You may ask in English, Spanish, and other languages. We would, however, really suggest, if possible, to write questions in English, because normally our experts speak the different languages you can see here, but you know, they are people as well, so they might be on holiday, on sick leave. And so finding experts who speak Polish or Italian, for example, is certainly much harder than finding somebody who can speak English and answer to your question in English. However, theoretically, we are open also to other languages. One last information is that all the information you share within the question you ask is treated as confidential, meaning it is not going to be shared with anybody else. And that sometimes is important with regard to novelty or registration of intellectual property rights. There are other also upcoming events, except for today. There's going to be actually quite large amount of webinar organized until the end of the first half of 2021, which for us is going to be on the 7th of July. So if you're interested in one of those webinars, please do not hesitate to register and you can take part to them very gladly. One last word regarding the ambassador scheme. I told you before, we've got ambassadors who are IP experts, part of the Enterprise Europe network all around Europe. So perhaps some of them are in the vicinity of your premises and perhaps might be very helpful to your activities and might be able to collaborate with you. They speak your language and they can also explain you better what the European IP Helpdesk is and does, and therefore it is our hope that they can be a valid help in your day-to-day activities. One last administrative information, is that the webinar today is going to be recorded, and the fact that the webinar that is recorded is only to allow you then to be able to see the slides and the recording in streaming after the webinar is over, or even if you have lost this webinar today, you are still actually going to be able to see it in another moment through the link that is going to be sent in the follow-up email. Those are a couple of further contacts that you will receive in the follow-up email later on, and starting from the website, and then the Twitter account, the LinkedIn account, and also our helpline account in order to be able to ask questions. One more information, I told you there is going to be a follow-up email that is actually true, and I'm going to send it to you at the latest tomorrow morning. We're just going to need the time to cut and prepare the presentation for you, and to prepare also the link for recording, and then send it to you. So in this follow-up email, I'm going to introduce also a couple of useful information regarding the webinar, and to help you navigate through the European IP Helpdesk infos that are currently published online. Finally, you might have already noticed that your microphone is turned off. So that means that you're not going to be able to directly speak throughout the whole duration of the webinar. However, that does not mean you're not allowed to ask questions. You're actually very welcome to do so, and as soon as a question pops out in your mind, simply write them, or write it on the right hand side of your screen. You should have a window that says questions. Simply write your question in there. I will gather them, and I will then open up a Word file later on after the webinar is over, where we will go through all the questions and try to give an answer in the time given. Now, I think I've taken already enough time of this webinar today, so I will simply give the word to our speakers today from the CPVO, who are Cécile Collonnier and Ursula Lamberti. It's a pleasure to have you both here. A very big thank you for my side, because I think I'll have the chance to learn something as well today, and I'm very excited for this new cooperation webinar with the CPVO. Thank you very much once again, and Ursula, I will leave you the word now.
Thank you, Michele. First of all, let me thank again. I know that I start every webinar like that, but thank you to thanks to the help desk for giving us a platform of discussion and to give us the opportunity to present our contents. This is a great collaboration, and thank you for this last webinar of the first part of the year. I take this opportunity to remind to our listeners that we are going to have more webinars in the second part of the year in about enforcement, R&D projects. So the help desk will keep you posted about that. Basically, I'm here today to provide you a couple of information on us, your speakers, and the Community Plant Variety Office, and explain a little bit why we are in a position to know something about this topic. So my name is Ursula Lamberti. I work as a legal advisor here at the Community Plant Variety Office, and I deal with legal aspects of protections of new plants, we can say. So the Community Plant Variety Office is a European Union agency, which means that we depend from the European Commission. We are financially independent, but we are under the umbrella of the Commission, like the help desk. Which means that the Commission gave us a mission, and our mission is to run a system of protection of new plant varieties, no? So innovation in the field of new plants. What we do is to grant a sui generis right on new plant varieties, but we also take care of the promotion of the system. We have a lot of international cooperation projects. We have an open dialogues with the industry, with other countries, with countries that are developing their systems. So we do not only grant rights, but we also take care of the promotion and the communication in this field. And so about new genomic techniques, we do not deal with them in a direct way because they are not part of the DUS examination, the technical examination that is the basis of the granting of the right. However, we are still working, especially thanks to Cécile, who has the knowledge in this field, in knowing more about these techniques and what are the future perspectives in this field. There is a lot going on. You will see that in the presentation, and that's it for my part. I will leave the floor to Cécile. Cécile is a technical expert at the Community Plant Variety Office. She has a very long-standing career in the research, especially in INRA, and she's our light when it comes to new techniques, molecular markers, etc. So I leave you in her very good hands and very knowledgeable hands, and I'll switch off my camera and maybe come back in case there are legal questions in the end. So thank you. So maybe Cécile, you can
try to share my screen now. Okay. So okay. So
Perfect.
Can you see?
We
Okay.
can see perfect.
Okay. Perfect. Oops. What did I do? Can you see it still, or?
Yeah, we can see it. No,
Oh,
not anymore.
Okay. Not anymore. And now?
Now we can see it, and I think, yeah.
But I can't. This is crazy.
We can see the presentation, but if you put it perhaps in full screen like you had it before, now we can see you.
Mm-hmm.
And
okay. Okay.
Correct.
I think that's good now.
Perfect. Yes.
Thank you very much. Sorry for this.
No problem.
So I'm very happy to be here today, and I will present to you the new ways of creating plant varieties, especially based on these new technologies of genome editing. And I will also talk to you about the challenges of their protection and their marketing in the EU. So the presentation will be structured in 4 parts. The first one will be dedicated to the presentation of the tools and their effects on the genomes, as well as their status for now in the European Union. Then there will be a presentation of their application to plant breeding, and then I will focus on the protection and then on the marketing. So first of all, the presentation of the tools. So as you all know, in the EU, all products that are or contain or are produced from GMOs must have an authorization prior to entering the market. And there's different texts that form the legal framework, the most important ones being the Directive 2001/18 and the Regulation 1829/2003. In the Directive 2001/18, an organism is defined as genetically modified if its genetic material has been changed in a way that does not occur under natural conditions. And there is a listing of the techniques that lead to the production of GMOs. And some of them so are considered as in the scope of the regulation, and some, however, they produce genetically modified organisms are considered outside the scope of the regulation. So here you have a summary of what is considered as out of the application scope. Of course, it's all the techniques that do not result in genetic modifications, like traditional breeding by crossing, in vitro fertilization or polyploidy induction. But you have also techniques that produce genetic modifications but are excluded if there is no recombinant DNA used and left inside the cells. There are mutagenesis and protoplast fusion. What is in the application scope is the transgenesis techniques considered as the classical ones, and also protoplast fusion if these protoplasts, of course, have been transformed before, so are already considered as genetically modified cells, and some old techniques that are a little outdated that were micro and micro injection and micro encapsulation of free DNA molecules. So things in 2001 were well defined, but since 2001, of course, the breeding techniques have evolved, and there's a whole set of new breeding techniques, the NBTs, that are now called by the Commission as novel genomic techniques that have been developed since 2001, so after the legislation was adopted. And in 2011, the Commission, to start clarifying the landscape of these breeding techniques, formed a work group that had the mission to identify these breeding techniques and list them. And you have them listed here. There are 8 of them. And so I will not read them all, but the 2 ones that are of interest today are the ones that compose the technologies that are considered as used for genome editing, which are the ODM, so oligonucleotide directed mutagenesis, and what is generally called targeted mutagenesis, based on the use of site-directed nucleases. So first thing, a little reminder of the definition. So the ODM technique is used to induce targeted single nucleotide mutations. So as you can see in the little drawing, the DNA, the double strand helix of the DNA is opened, and an oligonucleotide template containing the desired modifications that you want, so an intended mismatch is injected into the cell. And when this oligonucleotide template containing the modification is hybridized to its target, then there is different kinds of DNA repair mechanisms that will copy the change into the DNA, and then the template will be degraded. And at the next replication step, the intended change will be copied into the complementary strand. And so you you obtain a stable mutation. So what is a little particular in this technique is that the intended mismatch that you have put in the template is not always the one that will be copied into the genome, but at least you trigger a mutation at a very specific place. So this mutation can be a substitution of base pairs, an insertion of base pairs, or a deletion. And so this technology has been already used, for example, by the company Cybus for the production of herbicide-tolerant flax and oilseed rape by creating a point mutation in the ALS gene, which is a classical gene to create new herbicide tolerances. And the technology has been patented by Keygene, so it is called Keybase, and they propose a platform to produce traits. For the SDNs, you have 4 categories. Chronologically, the first one to appear was the meganuclease, and the last one, the CRISPR-Cas system. In between, you had the zinc finger nucleases and the talons. So this little table allows to present them through the main points, the main aspects in a simple way. So the meganuclease is a very big protein that is inspired from natural proteins that exist in living organisms. And so it's a protein that is symmetrical and that has in its center a catalytic domain that is capable of cleaving the DNA, the double-strand DNA. So you need only one big protein. For the 2 others, zinc finger nucleases and talons, these are purely synthetic molecules that do not exist in nature. They are composed of elements that were found in nature, but as such, they're completely synthetic. And so they are built in 2 parts. You have 2 elements that will bind to the DNA on both sides of the target where the cleavage will be done. And this cleavage will be done by an enzyme that is called the FOC. It's a bacterial enzyme, a classical one that is capable of cutting the double-strand DNA also. That has the particularity of being composed of 2 monomers that have to be close to each other to dimerize and then to be able to cleave the DNA. So that's why you have 2 elements bringing these 2 monomers close to the place to cut. So with the zinc finger nucleases, the 2 binding elements are composed of zinc fingers, and in talons, they are composed of synthetic tal that are elements that are present in some bacteria, pathogenic of plants, and whose code has been explained and that can be used to compose on-demand sequences that are specific to the target to cleave. And the last category is the CRISPR-Cas systems. So for this one, you need to have the protein that will cleave the DNA, that is the Cas protein, and it is guided to its target by an RNA that is then called the guide RNA, gRNA, and this RNA will be associated to the Cas before, so it leads us to the target site. So in terms of specificity, you can see that the off-target, meaning the capacity of the nuclease to cut not on the target but in another place of the genome. So with the meganuclease, the talons, and the CRISPR-Cas systems, it is rather low. The zinc fingers is a little less specific, even if there are some new strategies to try to improve that. In terms of diversity of target sequences in the genome, you have the possibility, it's a very, very large and so global average through the different species on which you can use these nucleases, but meganucleases can roughly find a cutting site every 250 base pairs, the zinc fingers every 500 base pairs. So it means that if you really want to cut a specific site in a defined gene, you may not always find a place to have your meganuclease or your zinc fingers active. But with the talons and the CRISPR-Cas system, the cutting sites are found every 35 base pairs or even every 8 base pairs. So it means that practically you can target almost every sites in the genome, every gene, every loci, provided of course that these zones are accessible and that the DNA is in a shape that allows the arrival of these nucleases. But it means that the CRISPR-Cas systems are extremely flexible, extremely useful to target all the sites in the genome. In terms of difficulty of production and price, you can see that the meganuclease is very difficult to produce and extremely expensive, and then it decreases in difficulty and price to reach a very, very easy and inexpensive system that is the CRISPR-Cas system. So it explains why, given all these elements, low of targets, large diversity of targets, easy to produce and very inexpensive. So it explains why the CRISPR-Cas systems has flourished and are now available in almost all labs if they wish to. So what these nucleases do to the genome? What type of modifications? So they arrive to the chromosome and they're gonna cut the two strands of the DNA. So they create what is called a DSP, a double-strand break, and this is recognized by the cell and has to be repaired quickly. And so there are different machineries inside the cell that can be used for that. So in the simple situation where you just have the break and you need to repair it without any other element, you don't give to the cell any template to help with the repair, then in higher organisms, the principal mechanisms that will be used is just a simple ligation. So its name is non-homologous end joining. So end joining says well what it means. You join the ends. And so usually this mechanism is pretty conservative and you repair the DNA exactly as it was. But the problem is that if you do that, the nuclease sees again the target accessible. So it's going to come back and cut. And so you have a cycle of cut and repair, cut and repair, cut and repair. And even if the machinery of the NSUG is very conservative, at some point a mistake will be done. And that's when you create a mutation. And this mutation then changes the sequence of the target, and the nuclease cannot bind anymore, it cannot cut. So you stabilize your mutation. And thanks to that, if you screen enough individuals, you find the mutation you want, and you can create the knockout of a gene. So it means that you stop the expression of these genes. You can create different types of mutations that can be deletions, insertions, and substitutions. And so if you have a break in the cell, and at the same time that you create this break, you provide a donor DNA that will serve as a template to repair the break, then you have either a direct ligation of this template, so you can see on the right hand, again, non-homologous end joining, and there is an insertion, a direct insertion by ligation. It's the frequency of this event is pretty low, and to help introduce the donor DNA at the break, what the scientists do is that they use a donor DNA that bears the gene desired, the gene of interest, that is flanked by sequences that are homologous to the target. And then, thanks to these homologies, the donor DNA will be integrated by homologous recombination, which is another set of mechanisms. So whether you use homologous recombination that will be done at a higher frequency than NSEG, or whether you use NSEG, you end by having inserted at the target the gene that you desired. And so for that, this action is called gene knock-in by opposition to gene knockout. So you can complexify things a tiny bit if you use several nucleases at the same time. So you can trigger the knockout of simultaneously of different genes at the same time. So this is easy, especially with the CRISPR-Cas system, because you have the capacity to multiplex the approach and to target, for example, polygenic traits. So you can target different traits involved in a given, different genes are involved in a given trait. For that you use the Cas, the protein that will cleave, and you direct it to different places by associating it to different guides. So if you do that, you can get rid of certain traits. You can also trigger chromosome rearrangements, so large chromosomal deletions or even inversions of clusters of genes. You can also try to have a multiple action by using only one SDN if this SDN has a target that is common to a set of genes. And that is very frequent in plants, that you have families of genes that are involved in the same type of function. And sometimes if you want to get rid of this function, it's very interesting to have an SDN that will target all these genes at the same time. And so you delete the genes, and usually, so in certain circumstances, these genes are put together in the genome and they make clusters, and so you can get rid of these clusters. So you can do a lot of things to modify the genome. You can also increase the efficiency of gene targeting. So gene targeting is the fact that you direct a genetic element, cassette, to the genome based on the homologies that you put on both sides of the gene of interest. So in higher organisms, the insertion of this type of cassette is very, very infrequent, very rare, because the mechanisms of insertion that is favored by the cells is usually DNA-HOG or other types of illegitimate recombinations. So to have a high efficiency of gene targeting, you need to be in an organism that favor homologous recombination. And they are very rare, these organisms. You have two examples that are almost the only ones. So it's Saccharomyces cerevisiae, that is the yeast of the baker, and a little moss that you can find in forests that is called Physcomitrella patens. And they are model organisms because we want to understand how they do that. But so the idea, the wish of the scientists was to try to build an environment in the cell that would lead to favor homologous recombination over illegitimate recombination. And using the SDNs is a good way to reach this goal, because when you create a double-strand break somewhere, it was already demonstrated in the 90s that the frequency of recombination and of gene targeting is multiplied by 10 or 100-fold in plants. So in the 90s, you didn't have the SDNs, so you knew you could improve gene targeting, but you could not cut where you wanted. You could not cut at the target. But when the SDNs arrived, then you had the possibility to really cut where you wanted and increase gene targeting at the locus that you were targeting. So the application of this gene targeting are multiple. So it can serve for fundamental research because it helps validating the function of the genes. So it's a support for functional analysis of the genes, and also it can be used for plant breeding for the modification of the genome with agricultural applications at hand. So different strategies can be used based on gene targeting. So you can do a gene knockout also. So you don't create a mutation like on the first slides. You insert something, but the presence of this insertion disrupt the gene. So in the end, the expression is stopped. So you do a gene knockout also by gene targeting. And you can also use something in another approach that is extremely interesting for plant breeding, that you can target a place in the genome, not to insert something or to disrupt something, but to replace the sequence. So without changing the gene, without changing the function, you can replace the allele that is present, so the nature of the sequence, by another one that you know is agronomically more favorable. You can also play on the regulation of the expression of the genes, because you can introduce different types of regulatory elements, like modifying the promoter. And you can also just continue to do classical transgenesis. Pursue the same goals as classical transgenesis, which is just insert a transgene that is completely exogenous to the genome to bring a completely foreign function. But this time, instead of letting the insertion happen completely randomly, as it is the case with classical transgenesis, you can really address your transgene exactly in the place you want in the genome. You control the transgenesis. The site-directed nucleases can also be turned into site-directed effectors. So it means that you use the elements that bind to the target, but instead of cleaving, you can bring regulators that are associated to these binding domains. And so you can do that with zinc finger and talons. So this time, because you don't need the FOC, the two monomers of FOC that need to dimerize, you just need to bring the effector. You need only one DNA binding domain associated to the functional effector. And you can also do that with the CRISPR-Cas system. And for that, you need to prevent the Cas from cutting, because that's not what you want. You just want to bring an effector that will do something to the genome, but without mutating the genome. And in that case, if you destroy so if you create mutations, if the two nuclease domains of the Cas9, you inactivate completely its capacity to cleave, and you make what is called a dead Cas9, the dCas9. So once you have the dCas9, you can use it to bring to a certain place in the genome something that is attached to her. So the guide associated to the Cas will bring the Cas to the place, and the Cas will have a little hat, something that it will just be put close to the target for the effector to have an effect on the target. So these different functional effectors can be used to regulate gene expression positively or negatively. It can also be epigenetic enzymes or recombinase or transposase. So you can trigger at the target a modification of the gene expression, a recombination, or a transposition. And I'm going to finish this overview of the different tools by mentioning the latest ones arrived that are base editing and prime editing. So base editing is based on the use of a dead Cas9 that is fused to a cytidine desaminase. So you can see on the little drawing that the guide RNA, the little buckle that brought the Cas9, this little brown potato, to the target, is associated, you can see it to the bottom of the drawing, to the cytidine desaminase. And so this cytidine desaminase, being in the vicinity of the genome, will do what it is made to do, which is modify a C base into a U base. And it will do that only on one strand. And so the cell has to wait for the next DNA replication to transform this U into a T, and then to have also a modification on the complementary strand to have the corresponding base. So that's done by the DNA repair machinery. So this cytidine desaminase can trigger a punctual mutation at a defined place that you can choose by the place where you're going to fuse it to the Cas9. And so it is a good way to have a mutation where you want without risking any off targeting, because as you don't cleave the DNA, the chance to have a mutation made in a place that is not the target is very reduced. And for prime editing, you use again a dead Cas9 that will just serve as a holder of the effector. And in that situation, the effector is a reverse transcriptase which is capable of triggering a cleavage in one strand of the DNA, then copy the sequence of the guide. So use the guide as a template to recopy inside the genome the sequence of this guide, and then there will be different steps so that will be done by the DNA repair machinery of the cell and by the retrotransposase to create a little cut in the opposite strand and copy the sequence of the guide that was already inserted on the first single strand. So it's a little complex, but what it does is that in one go, you have a unique cleavage on a single strand that allows to insert a sequence that is bared by the guide. So as the guide must hybridize to the target, when you use that, it's mainly to replace an allele, because the basis of the sequence of the guide needs to be, and almost all of them needs to be homologous to the target to hybridize, but you can introduce a few mismatches that corresponds to the mutations that you want to insert in the genome. So that's another way of creating mutations. I just wanted to mention it because it's a new one that will probably be used soon in plants. For now, it's only on animal cells. So what are the applications of all these capacities of the gene editing technologies to plant breeding? So I wanted to start by presenting them, reminding you that finding a given mutation in nature is just a matter of time, because mutations are triggered in the genomes absolutely all the time. And it is essential to evolution. And I put two examples on wheat and on maize. At each generation, in 1 hectare of wheat, you have all the genes showing at least one spontaneous mutation. And if you look at a set of kernels of grains of maize, you can be sure that in each of them, there is 20 spontaneous mutations that make them differ from their mother plant. So creating mutations is something very current in nature and absolutely systematical. But if you want to detect the mutation that you need, and maybe the set of mutations that you need, then it can take a lot of time. And if you want, for example, there's a little exercise that was done at the bottom of the slide. If you want to find in tomato a locus where 2 specific mutations will have happened, these mutations, for example, are capable of giving some tolerance to viruses. So these mutations, you saw them in pepper. Tomato has a similar gene where these mutations could occur, but to be sure to see these 2 mutations at the same time in an individual, given the different parameters of the tomato culture around the world, we made an estimate that you could have to wait for 120,000 years to see an individual, and in the situation where you would screen all these individuals on a planet. So looking for a specific mutation can take a lot of time if you wait for nature to make it. So you can accelerate things by doing in vitro random mutagenesis, which is a technique that was developed in the mid-20th century and that was used since then really intensively. There is more than 3,200 varieties in the FAO mutant variety database, which represents more than 200 species that were treated either by radiations or by chemicals like EMS to trigger mutations randomly in the genome, and they were then selected to create new varieties. So I put some examples, for sunflower with a high oleic content, the grapefruit with red flesh, or self-compatible cherry tree, but there are many, many others. So if you look at this graph, you can see on the left side that when you use an SDN, when you do gene editing and you are in a situation where you control the off-targeting, you will be in capacity to identify individuals where you have only one mutation exactly triggered at the locus targeted. So of course, you can see in the middle of this scheme that you will always have all the spontaneous mutations dependent on the different species. And at the left hand, you see what happens when you use this in vitro random mutagenesis technique, which is, for example, here in that case, just soak the cells into some EMS. So in that case, for tomato, in average, for every individual, you will have, because of this immersion in EMS, 600 mutations. And so if you want one or few, you have to get rid of all the others. So you have to backcross a lot and do a severe selection. So in the end, it's to illustrate the fact that gene editing has the advantage of allowing the creation of desired mutation in a very fast and a very controlled way, well, compared to other types of technologies. So when you use the SDNs, you can trigger mutations in a very controlled way, just like I said, and you can also give a donor DNA that will be inserted at the target. The donor DNA bringing only a few mutations because he's almost similar to the target, or bringing a completely new gene, an exogenous element to the cell. And these three categories of events were defined by EFSA to clarify on the discussions and also be able to take decisions on a case-by-case basis. So the first one, when you create just point mutations, one or a few, you create products that are called SDN1 products. So with base and prime editing, you also create punctual mutations that are of the same type and that can be put in the category or associated to the category of SDN1. When you present a donor DNA that is almost similar, except for a few mutations, and you are doing a replacement of the allele targeted, then you produce SDN2 products. And when you insert a completely exogenous transgene that will stay there, then you produce SDN3 products. So I'm going to illustrate how you make these three types of products, the SDN1, 2, and 3. So SDN1 can be obtained either by a stable or transient transformation technique. So in the stable technique, you can use agrobacterium, and you can see that so you need first to insert the sequence that will code for the nuclease for it to be able to create the mutation in the cell. So once it's done, you have the mutation that you desired, but you still have the sequence coding for the nuclease. And you need to get rid of that, because if you leave it there, you're going to have probably some off-targeting arising. And also, you don't want to create, you don't want to have a real transgene in your variety. So in plants, it's pretty easy because you start some backcrossing, and you're going to identify by selection individuals that will have got rid of the sequence coding for the nucleases, keeping the mutation. If you do a transient transformation, it means that you transiently propose to the cell what is necessary to create the mutation, but there is no sequences coding for these nucleases inserted into the genome. So you don't need to backcross. You have directly the mutation you want. Voilà. You can do that either by bringing some sequences that will code but without being inserted, or also directly by bringing the nuclease in the form of the protein. So for that, you can use either a protoplast fusion or bombardment by particle gun. So for the SDN2, it is exactly the same thing. You can use stable transformation or transient transformation. But the difference is that because you bring a donor DNA, when you do a stable transformation, you will have to insert somewhere using a classical transformation technique. So this cassette will be integrated randomly. You have to insert somewhere the sequence coding for the nuclease, but also the donor DNA. The nuclease will cut the target, and you will have put the same sequence on both sides of the donor DNA to have it released at the same time of the cleavage and be available for the repair. And so once again, you have to get rid of the sequence of the nuclease by backcrossing. If you do that transiently, you have to bring the elements allowing to have in the cell at the same time the nuclease and the donor DNA, but without any insertion. So you don't need to do backcrossing. For SDN3, exactly the same issue as SDN2. You have to have the sequence coding for the nuclease and the donor DNA. The difference is that when you do SDN3, you look at the possibility to introduce the donor DNA into a specific place. But for that, you need to have identified this specific place. So people that want to do to follow the SDN3 strategy have to find in the genome what we call landing pads, so zones in which the donor DNA can be inserted. And these landing pads are characterized and selected because they have a good environment, a good genetic environment, allowing a good expression of the donor DNA and good stability of the insertion of the donor DNA. So now a few examples of plants modified according to the three strategies, SDN1, 2, and 3. So for SDN1, I took an example of a plant modified with talons. So by bombardment, particle gun, some cells from bread wheat were transformed with talons to create mutations. And so the particularity of this example is that bread wheat is an hexaploid, so it means that the target existed in the genome in 6 different copies. So there were 6 alleles to knock out, and that was successfully achieved by this team because they managed to create mutations. So not always the same ones, but at least they were all knocked out in the 6 alleles encoding for a protein that is involved in the resistance to mildew, and they produced mutant lines with a strong resistance to powdery mildew. For SDN2, I took the classical example of the production of herbicide tolerance or resistance. So this example is in tobacco, and it was the mutation was created in the gene using zinc fingers. For SDN3, I took an example in cotton using meganucleases. In that case, so they wanted to do the stacking of new genes with already present transgenes in the variety. So they were somewhere in the genome inserted by classical transgenesis, already 2 genes involved in herbicide tolerance, HPPD and EPSPS, and they wanted to, there were already 2 genes that were a BT gene and a BAR gene, so resistance to a herbicide and to insects. And they wanted to introduce next to them these 2 genes, HPPD and EPSPS. And they managed by having a meganucleases that was specific to a site that was close to the already present transgenes, and they could have the insertion of the donor DNA bearing the 2 new herbicide tolerance genes next to the 2 previous ones. And so it's interesting to do that because if you want to use these individuals in selection schemes by crossing, then you have all the genes inherited as a single genetic unit. All the genes are linked together. So basically, almost all characters for which the genes involved are known, can be modified by gene editing. But so it seems very straightforward, very easy, but it doesn't replace all the years of study that you need to identify these genes that you want to mutate or you want to replace. So you need to go through QTL's analyses and all the kinds of new genetic studies now to identify candidate genes. But to accelerate their validation, you can use the SDNs already, because you can trigger knockout of these candidate genes and confirm their implication into a given function. So you use the SDNs to validate your candidate genes, and then you can use them again to integrate these interest genes into your genome. But that takes a lot of efforts. So I listed all the applications, all the traits you can think of, just to illustrate the diversity of the possibilities opened by gene editing. So you can create disease resistances or tolerances, herbicide tolerances. You can modify the anti-nutritional compounds and allergens in the varieties. You can improve the nutritional value. You can also modify some of the components of the cells, like starches or fats for food and non-food uses. You can extend the shelf life, reduce the wastage, reduce the enzymatic and non-enzymatic browning. You can also improve the yield by playing all kinds of different enzymes involved in photosynthesis and the conversion into organic compounds like the riboscope genes. You can also improve the biomass conversion for biofuels by working on the lignin. And you can also, and it's very interesting for breeders, control meiosis and more specifically increase crossing overs or produce diplogametes for the fixation of certain genotypes. So there is a very high potential of genome editing for plant breeding for additional reasons that I listed here. So the first one, of course, is the fast assembling of alleles and genes that you all understood, but it is, I want to trigger your attention on the fact that it is especially useful for species with long production cycles, because you can modify the genome in less than 5 years and already you can be in capacity to observe already the effects, so you can really speed up the selection schemes for woody trees, fruit trees, etc. You can also found gene of interest in other species and access them, have the possibility to adapt them, to integrate them into crop species. So you have an access to a wider range of species, and you can use a wider part of the existing genetic diversity. You can also mount programs for minor or orphan crops, because as you reduce the time of the selection schemes of the breeding work, this type of investment starts to be cost effective, and you can get some return on investment in an easier way. You can also have families of varieties with the same original genetic background, that is of high agronomic value or high environmental value, and you can then adapt this original variety by really fine-tuning the genes in the genome. You can adapt it to very specific environments. So you can design varieties that are adapted to very local conditions and create portfolios of families of varieties that the users, the farmers, will then just be able to select really precisely according to the conditions in which they work. And you can also use genome editing to increase genetic diversity. And there's two concepts that I will present to you: the rewiring concept and the induction of targeted genetic variability. So the rewiring concept is it's based on the fact that during the domestication and then the modern selection, we maintained in our varieties beneficial mutations, and we selected new beneficial mutations according to specific conditions at the time they were produced. And sometimes we didn't pay attention that we left in the genome, and we couldn't know at that time, but so we left in the genome undesired mutations that were not selected because we were not in the conditions where they would appear as detrimental. So now that we have conditions of culture that are evolving, we are in stress conditions more often, we start to see that the modern varieties can have so we reach the limits of their capacities. They're blocked by the presence of all these undesired mutations. So the idea of the rewilding is, because now we have the capacity to identify all these genes, is to see what were the alleles in the original wild plants for this loci, and then copy them into the modern varieties, keeping all the beneficial mutations accumulating and getting rid of the undesired ones. So we will improve. So there is the objective of improving the modern varieties by mimicking the alleles present in the wild parents that resisted better to the conditions of stress that we are now facing. The targeted induced genetic variation is a very simple way to create diversity if you know a gene that is involved in a specific trait of interest for you. So for example, here there is a gene, so ELF1, involved in the time of flowering, and so you screened your germplasm, and you know that you have different alleles available, but they do not present the genetic diversity that you would like to have. So what you can do, you produce protoplasts, so you isolate cells without their walls, and you cultivate them in presence of a nuclease. For example, here with the CRISPR-Cas system, you add the Cas9, and you add a series of guides that will guide this Cas9 to different targets, different places inside the gene. And so you know that because of the system, you will trigger in the different cells, because it will act randomly, you will trigger the cutting, the repair, and then the appearance of a mutation. So a mutation at the different places targeted by the guides, the guide RNAs, and these mutations will be random themselves, so there could be insertion, deletions, etc. So you will have in all these cells this gene mutated in different places, and so the different mutations possible will be also very diverse. So if you regenerate from these individual cells, individual plants, then you can screen them in the greenhouse to detect if the phenotype of, so the time of flowering was modified by all these mutations. So it's in that case, the trait is very easy to see. I mean, if you want to have an early flowering, you will very easily see the plant that will start to flower before the others. Then you recuperate this plant, and you can sequence the gene ELF1 and identify what mutations triggered this new phenotype of flowering. So that's a way to increase the diversity, the genetic diversity for specific genes involved in traits of interest. And because you can multiplex, I told you, you can have the Cas9 with a lot of different guides. Here they were all targeting the same gene, but you can also use guides that will target different genes at the same time, and then you really start to reshuffle the sequence of the genes, and you can produce very new phenotypes. So concretely on the market, what are the new edited varieties? So with talents, it's Calixtan was producing them. You have already on the market a soybean that is high oleic and low linoleic acid. So it's already in production in Argentina and in the USA. You also have potato where some enzymes have been modified to reduce the content in acrylamide in the tubers in certain conditions of storage. And so these potatoes are still, so they are ready for production, but so the Calix is starting to pass the contracts with the farmers for the production. We're really close to have them on the market. And for other products like low saturated canola and gluten reduced wheat, the projects are ongoing. This is still not in the phase of production, but we already have edited varieties based on the use of talents. For the CRISPRs, so there were some proposal made by different companies that tested in America the possibility to put these varieties on the market without them being regulated. So that was the case of Pioneer that asked the USDA to if they would consider as regulated or not waxy corn hybrids that would be developed using CRISPR-Cas. So there is already a lot of waxy corn on the market, but with I mean using the CRISPR-Cas, they were hoping to create mutations that would so block the production of amylose, but without impacting the rest of the genome, and hopefully leading to a higher yield because it can be a little low in waxy varieties. So DuPont Pioneer published also other types of research that they performed on maize to improve the yield under drought stress conditions. So they did that by working on the promoters of certain genes. And here you can see two examples. They added an additional promoter, and they also swapped an existing promoter. So they did like an allele replacement, but as it is for a promoter, it's not an allele replacement sensu stricto. But they managed to have good results. And so varieties with this type of modifications are in the pipelines. So another product that is already available is the famous white mushroom that never gets brown, because the professor at the Pennsylvania State University who produced it targeted, so created a mutation in a polyphenol oxidase. This product also would be considered as not regulated, so it's not really on the market, but the professor is open to sign any contract with somebody who would want to initiate the production. There is also a more recent event concerning tomato for gardening, edited by CRISPR-Cas. So it was produced by the University of Tsukuba in Japan, and it will be commercialized by Sanotec Seed. So what they did is that they increased by 5 times the amount of GABA, which is a major which has a major role in neurotransmission, and that so has been identified as being effective in reducing blood pressure and relieve stress. So it's a distressing tomato, and that's the first edited variety that was authorized by Japan in 2020. So that's all for the little overview of all the products that are already available. So you see that they're close to arrive on the market, but it's just really the beginning. So something that can explain that this type of varieties arrive only now on the market is a little delay in the initiatives at the origin of the creation of the tools because of their IP situation. So for talents, it was pretty clear. It's Calix who has the rights. and will give licenses. For the meganucleases, there was a little battle between Selectis and Precision Bioscience, but now it's over, and they have agreement on cross licenses, etc. So the situation is pretty clear if you want to use talons and meganucleases. But for the CRISPR-Cas system, the situation is really not clear, and that's why the breeders were a little hesitant at the very beginning of the arrival of these tools on the market. So most of them, they started to work with them, but being prudent, because they didn't know with whom they would have to get agreements. So it comes to, just to tell you in passing, the story of the CRISPR-Cas system has started long before the Doudna and Charpentier's work. So there were a lot of studies initiated starting from the 90s by many microbiologists, and so it's in certain, now some of them are receiving a little acknowledgement of all the work they had done to allow Doudna and Charpentier to make this fantastic discovery in 2012. So regarding the IP, Doudna and Charpentier so got the first patent with claims on the activity of the system from a bacteria called Streptococcus pyogenes, but without really mentioning that without demonstrating that the system was applicable to eukaryotic systems, eukaryotic cells. So it means that they thought they were covering all the applications, but the first one that really declared having tested the CRISPR-Cas system on eukaryotic cells, so on animal cells, is the MIT with Feng Zhang. And so they got a patent on the use of the same system from the same bacteria on eukaryotic cells, considering that the first patent was covering only prokaryotic cells, meaning bacteria. So superior organisms were covered by the MIT, the Broad Institute patent. They also got, just in passing, another patent on another nuclease that is not the Cas9, but the CPVF one that has now a new name. But so they were very active in this area too, and you can see that they got the patent on animal cells just a few months after Doudna and Charpentier got theirs on bacteria. And so to avoid any confusion and to counter the patents claiming that the MIT had really discovered the system and applied it to animal cells, the University of Berkeley required an interference procedure so related to the patents held by the Broad Institute and the MIT. There was another actor involved, which is the Vilnius University from Lithuania. They were working also at the same time, and they got also during the same year and even before, and the patent of Doudna and Charpentier, a patent on the CRISPR-Cas9 system, but this time from Streptococcus thermophilus and not Pyogenes, and also covering the version that can trigger cleavage only in one single strand DNA, so the Nuclease version. They also had worked on a system for cleavage on RNAs, which is very interesting now. So nobody started anything, but the USPTO in 2016 identified a potential interference also between the MIT and the Vilnius University. So what happened is that in 2017, the interference procedure failed, and the USPTO upheld the patents of the Broad and said that they would stand. So of course Berkeley appealed, but the Court of Appeals denied the request in 2018. So Berkeley refused. So there were a lot of discussions with the Broad. Broad proposed to create a patent pool and placed new claims upon the patent trio, but Berkeley refused this offer from the Broad. They appeal, And finally, the interference was declared with the broad patents in 2019. So there were a hearing that started in 2020, and the broad, well, no, what do you want? So yeah, the patent office decision declared the broad as the senior party because of a priority date of only 47 days before before Berkeley. So each party were invited to present evidence, so lab records to establish the actual date of the invention, and they had until May this year, and now there's an oral hearing ongoing. So as you can see, I mean, a long story, just to demonstrate that the situation is still unclear, and well, it's not over yet. It didn't prevent the Nobel Committee to give the Nobel Prize in Chemistry in 2020 to Doudna and Charpentier. So there is some kind of recognition of their work already. Okay, so now we move to the protection of the innovations related to genome editing in the EU. So the opinion G319 of the EPO Enlarged Board of Appeal in 2020 on paper confirmed that the exception to patentability of essentially biological processes for the production of plants and animals has a negative effect on the allowability of product claims or product by process claims directed to plants, plant material, or animals if the claimed product is exclusively obtained by means of an essentially biological process, or if the claimed process features define an essentially biological process. So the EPO proposed that the claims to technically mutated plants or animals of applications with priority or filing dates are on or after July 2017 will include a disclaimer to exclude plants or animals with the same characteristics and having been exclusively obtained by means of an essentially biological process. So all that means that there is a possibility, thanks to this disclaimer, to freely make use of the same mutation if it is found in a plant where it occurred spontaneously, meaning exclusively by means of an essentially biological process. So what about the allowability of a technical step that is kind of mimicking an essentially biological process? For that, the opinion of the case G319 confirms the previous case law, saying that it is not irrelevant whether a step of a technical nature is a new or known measure, whether it is trivial or a fundamental alteration of a known process, and whether it does or could occur in nature. So it means that when you want to patent a product so that you made using random mutagenesis, it is patentable, even if spontaneous mutagenesis can occur in nature and could have produced exactly the same thing. And what about the allowability of products bearing sequences that are already identified in nature? Because, okay, you can use a technology that mimic nature, no problem, because you create something new. But if this thing is already known, has been already observed in nature, what happens? For example, if I take back the example of the genes with the 2 mutations found in pepper to get a tolerance to proteoviruses, and you want to reconstitute them into the tomato genome, like it was done in the framework of the Genius project in France, can you patent that? Because these sequences were already described. And to be patentable, plants have to be novel and inventive. So if we try to put that in writing, all the products or product by process claims, depending on the techniques used, are you patentable or not? So here are listed the techniques that really leave recombinant DNA inside the cell. So the classical transgenesis, that can be intragenesis or cisgenesis, depending on the elements you use. And then the SDN3 strategy, that is the directed mutagenesis, so the directed insertion of donor DNA. So all these techniques, for all these techniques, the process is a technical step. It is not an essentially biological process. So you are eligible, you're not excluded from patentability according to the Article 53B of the EPO law. So, but if you look at the products, if you use synthetic sequences, you can legitimately be considered as not excluded from patentability. But if you use native sequences, like it is the case for cisgenesis, or it can be the case for SDN3, then what about your allowability regarding the novelty, the Article 54? Is it really irrelevant whether it could occur in nature or not? So that's the difficulty for the examiners. They have to identify if they comply with these requirements or not. And for the other types of techniques that are the SDN2 and ODM and SDN1, we're still in the situation of a technical step, so not excluded from patentability according to Article 53B, but the products can vary, and then the same questions arise. So you can use SDN2 and SDN1 and ODM to create mutations that are not yet found in nature. So at some point there will be, and for that the disclaimer is very useful. If you find later on a mutation that is exactly the same, thanks to the disclaimer, you can use these plants without owing anything to the owner of the patent. But if you directly want to create plants with where the desired mutations exist in nature, then you have to ask yourself the question, and the examiners have to decide if you're really excluded or not. And for random mutagenesis, it is the same questions. So whether if you use something that is not yet found in nature, you have the disclaimer. If you use something that exists in nature already, are you really eligible? And for spontaneous mutagenesis, this is the only case where you are in a situation where it is an essentially biological process that triggered the random mutations. And in that case, because you don't have a technical step, you are excluded. Regarding the number of patents based on the use of the CRISPR-Cas systems, there is a very interesting publication in Nature Biotech that was published in 2019 that made a survey of all the patents and identified that for 15% of them were dedicated to plants, and that the main actors in agriculture were definitely China, with 259 CRISPR-Cas patents, and then America and Europe in a very much more modest way. So how to protect plant varieties in the EU? Because the decision G319 confirmed the non-patentability of plant varieties. So it means that if a plant variety is not identified in the claims of a patent, the scope of the protection will not cover any specific variety. And in principle, the invention can be applied to an indefinite number of plant varieties, such that several varieties can be covered by the claims, but not protected for themselves. So if you really want to protect these varieties for themselves, then you have to go through our protection system in the EU that Ursula already evoked, that the CPVO is in charge of implementing. So this system grants IP rights according to the principles of the UPOV Convention. So it gives an exclusive right of exploitation to a variety during a given period that is usually 25 years, but that is extended to 30 for trees, grapevine, and potato. And this plant variety rights are different from patents in different for different reasons, but mainly due to the fact that the UPOV Convention defines the breeder's exemption, and this exemption authorizes anybody to use a protected variety to create a new variety distinct from the initial one, without requesting the authorization from the owner of the initial variety. So these rights are given to all species and genera. So far, we have 2,000 species covered, and the rights that are given are valid throughout the whole European territory, according to the principle of one key, several doors. So what about the testing of these edited varieties? What does it change to the DUS testing that is necessary to base the decision of granting some PDRs? So DUS, just as a reminder, it means distinctness, uniformity, and stability. So the new varieties needs to be distinct from all the previous ones. They need to be uniform and stable through their progenies. And this DUS tests are mainly done in the field or in the greenhouse. And so it means that we have to put the plants out. So if there are GMOs, then it triggers some questions. We're going to see that in the last part. But for now, just for the evaluation of the DUS character, so the DUS status of the edited varieties. So it shouldn't change much whether you are an edited or not, an edited varieties or not, except in the sense that if you're an edited variety so far, you have to include in your application file a declaration, because you are considered as a GMO according to the Directive 2001/18. And so the problem is that there is no detection possible, there is no control possible. So the system is a little jeopardized, let's say, by the fact that now application files have to be filled in and read really in an atmosphere of trust and in between the partners, but without any means of control. So for edited varieties, the evaluation is based on a complete loyalty between the partners. So without controls, the loyalty is always very difficult to maintain. But the gene editing technologies are used to breed. So it means that they're going to introduce traits into varieties. And so it may change something to the DUS testing if these traits are not in so do not belong to the list of characteristics that are evaluated according to our technical protocols. So if there are traits that are just modified in their expression, but that do belong to the technical protocols, nothing is changed. We don't want to know. But if the traits that are brought by these modifications, these additions, are specific characteristics that are not part of the protocols and are on which, so they are the characteristics on which the breeders decided to protect the variety and to distinguish it from the other varieties already available, already known. Then these characteristics have to be treated as additional characteristics. So they have to fulfill all the UPOV requirements, but they necessitate the setup of specific tests. Then, based on these results, the distinctness is established using the same minimum distances as usually. So apart from having to set up specific tests, basically, the current DUS procedure should not be fundamentally modified by the arrival of edited varieties on the market. So we should work approximately the same way, but we may have to decide if we accept all the additional characteristics that are proposed and if we are in capacity to evaluate them. However, gene editing could have a significant impact on the UPOV principles, and especially on the breeder's exception, because if new edited alleles are patented, which there will probably all be, then their accumulation inside the genome may reduce the access to these genomes, to the genetic backgrounds of varieties. Because if you start to cross a variety that is stuffed with protected alleles, then it's really a nightmare to get rid of them. And also, why do that? Because these alleles are probably the reasons why you are interested in this variety. But the problem is that if you keep these protected alleles, then you owe something to the patent owner. And so if these new edited alleles are not patented, then they could easily be copied in other varieties, even without the need to use the breeder's exemption itself, because you don't even need to make any crossing. You identify, you sequence the variety, you identify the allele of interest, and then you just copy it in your own allele lines. So it triggers a lot of questions on how to make value of the efforts produced by the initial breeder. So there is also questions related to essentially the derived varieties. So this essentially derived varieties, just as a reminder, are varieties that were identified after creating or observing a modification into an initial variety. And this variety that is essentially derived must be clearly distinct from the initial variety, but also conform to this initial variety in the expression of its essential characteristics. And if you find an EDV, the commercialization is authorized only if you have the consent of the initial breeder. But so what you need to remember is that the ADV is defined by the expression of the essential characteristics. So genome editing will may have an impact on that, because if you imagine that you successively edit the same elite line and you see all this mutation in red, they start to accumulate. So I mean, in terms of proportion in the genome, it's really infinitesimal. It's very, very small. The genetic background remains the same, but you start to accumulate a lot of work to have modified all these different alleles. So how many mutations do you need before a variety is not an EDV anymore? The question can be asked. Is there a need for an adjustment of the similarity threshold that are used, or is it desired to keep the scope of the EDV as it is, meaning that the initial variety will cover all the derived ones, no matter how many efforts of mutations you deployed? And so regarding this essential characteristics, there is a very easy way to escape this condition, because if you know the genes involved in these essential characteristics, by triggering targeted mutations, you can very easily not be considered, if the regulation stays the same, as an EDV. So managing the EDVs in the context of genome editing is much more complex than with classical transgenes, because just a few mutations can lead to escape the status of EDV. So it's something that breeders and UPOV are actually, and all stakeholders are tackling right now with the revision of the EDV concept, and there are discussions ongoing at the UPOV level. Regarding the marketing of edited plant products in the EU, since July 2018, all edited varieties are considered as GMOs, and so, as I mentioned already, they have the breeder that will want to have his variety listed will have, just like for protection, to provide Part B of the notification dossier for the authorization of the release in the environment. So this Part B is an authorization to set up trails, and is absolutely necessary so for the national listing. The fact that they are considered, that edited varieties are considered as GMOs in Europe makes things a little complicated at the world level regarding the exchange of goods, because you have different regulations depending on the countries. You can see here the countries in green were genome edited crops are not regulated automatically. And in yellow, those who are thinking about this issue. But so the question is open on the impact on world trade. So the fact that all edited plants are regulated in Europe may create really some difficulties for the big companies, the international companies. So the question was asked because of this situation of the legitimacy, the adaptation of the current European GMO regulation for edited varieties. It is extremely time-consuming and extremely costly to generate the data that is necessary to request a release on the EU market. And so we need to find an appropriate level of regulatory oversight. It is absolutely crucial to get all the benefits from these technologies in plant breeding. And so, so the regulation is needed if we think there are risks. So what does gene editing change in terms of risk assessment? And so this question was asked to the GMO panel of EFSA, and it issued different scientific opinions on the adequacy of the existing guidelines for the risk assessment of NBTs, and more specifically for cis and intragenesis, ODM and SDNs approaches. So the result, what's published in their opinion is that the current guidance for SDN3 products is perfectly applicable, and that on a case-by-case basis, even lesser amounts of even specific data may be needed. So they open the door to some flexibility in the data requirements for risk assessment. And for the SDN1 and 2 and ODM, they did not identify new hazards connected to these technologies. They declared that the guidance were so applicable, and they added that it was only partially applicable, but only to say that it was too much, that because of the absence of exogenous DNA in the plants produced by these strategies. So it is perfectly applicable, except that it is asking too much, so it should be reduced. In terms of detection, so that's really the center of the difficulty to implement the regulation. The detection for any GMO is legally required, but once you have mutations that you can find in nature, for example, after some time, the detection method that you provide to detect specifically the event that is in your variety is not specific anymore of your variety. It is specific of a given sequence, but not of the variety. And so for SDN products, so because what is inside the genome is an exogenous DNA where there is enough specificities to adapt a detection method, it will remain relatively easy, except that in the case of the stacking at landing pads, we're going to lose the signature of the events that was used before. That is the fragment located at the junction between the transgene and the rest of the genome, because now we control and we can place all the transgenes in exactly the same place. With the classical, the conventional GMOs, as the insertion was random, the place of insertion was really the signature. So we lose that, but it should still be feasible. But for ODM, SDN1 and SDN2, it's extremely challenging because the differences so corresponds to only a very limited number of nucleotides in a whole genome. And so if you don't know what you're looking for, it will be extremely difficult to find it through all the sequencing data that you can produce. And also it will be anyway impossible to discriminate these mutations from the same mutations obtained by other techniques. So the difficulty of the detection, that is sometimes almost impossible if there's no declaration, lead to the fact that the regulation cannot really be implemented. The implementation cannot be guaranteed. And so what's the point in having a regulation if it's not implementable in all cases? So All that was stated by the group of chief scientific advisors, and they advocated for the revision of the GMO directive. So a re-evaluation of the regulation appears necessary. So in 2019, the Council, based on this situation, requested the Commission to submit by April of this year a study regarding the status of the NBTs, now called the Novel Genomic Techniques, under the Union law. And they also asked the Commission to submit a proposal, if appropriate, to progress on this issue, or otherwise to inform on the other measures that would be required to continue in this topic. So the study was published on time. It gives a state of play on the implementation and enforcement of the GMO legislation as regards the NGTs. That was based on the contributions from targeted consultation of the member states and the stakeholders, and based also on the work of the ENGL and the European Network of GMO Laboratories, together with the European Reference Laboratories, the GRC, on the detection of products obtained by the new mutagenesis techniques. The study gives also an information on the status and use of the NGTs in the different organisms and for different applications, an overview of the risk assessment, also of the current and future developments, and an analysis of the ethical and societal implications. The conclusions are that there is a limitation in the capacity of the legislation to keep pace with the scientific developments, and this causes implementation challenges and legal uncertainties. So the second conclusion stated that there are strong indications that the applicable legislation is not fit for purpose for some entities and their products, and that it is not justified to apply different levels of regulatory oversight to similar products with similar level of risk that were obtained either conventionally or using entities. The third conclusion is that the follow-up of, so there should be a follow-up of the study. It should confirm whether adaptation is needed, and if so, what form it should take, and which policy instruments should be used in order for the legislation to be resilient, future-proof, and mainly uniformly applied. The fourth conclusion is that the study has confirmed that NGT have a very high potential to contribute to the agri-food systems in line with the European Green Deal, the Farm to Fork strategy. So we should have a policy action that should aim the use of these NGT products to contribute to sustainability, but without impacting the other agricultural sectors, and especially the organic one. And finally, the future policy action will also need to address the knowledge gaps and limitation identified in this study, because there's still a lot to explore and to clarify. And so more effort should be made also to inform and engage with the public on NGTs and all their views. So the next step will be that the Commission initiate a policy action on plants produced by targeted mutagenesis, but also on cisgenesis. They're going to include that into this initiative. And so this policy action will involve an impact assessment and a public consultation. And it will aim at finding a way to proportionate regulatory oversight. And of course, one of the biggest project is to engage a wide-ranging communication effort to share the results and to discuss the outcomes with all the stakeholders. In conclusion of this presentation, if you want to summarize, genome editing technologies are very valuable tools for plant breeding. I hope I illustrated that correctly. There are still some limitations to their use. They are due to the still unclear situation about IP and CRISPR-Cas system, always the wild card of the public perception, on which there's a lot of work to do, and also the legal status in the EU. But as you saw, potential evolution of the regulation may be envisioned. And regarding protection and marketing of gene-edited relating plant innovation in the EU, it requires the definition of appropriate IP strategy, taking into account for patenting the issues connected to the EBP and the novelty of the especially concerning the native sequences, and for listing and granting of CPVO, the necessity, always so far, to go for a GMO notification. I thank you for your attention, and I'm open to questions now.
Thank you very, very much, Cécile, for the very interesting presentation. And as I was introducing before to all participants, you are free at any moment to ask questions. Simply write them down in the question part of the screen. You can already write them, and somebody has actually already written the first one, but it's more for me, Cécile, and not for you, so I'll take over the answer to this question, which I will show them now. So Janine, Janina has asked if it's going to be possible to receive the presentations, and is also congratulating you, Cécile, for how the presentation were looking amazing. And the answer to this is yes, you're going to receive the follow-up email at the latest tomorrow morning. Just let me take control again, and you should be able to see the questions now. Yes, perfect. So when will you receive today's slides and recording? That's going to be tomorrow. And then there's okay, the second question from Professor Cécile is again to have access to the information. So again, I will provide you with a follow-up email. Don't worry about that. Everyone is going to receive today's presentation. Actually, also the ones who registered for this webinar but were not able to attend. And in case you should have further questions after the webinar is over and you didn't really manage to ask those questions during the webinar, don't worry about that. Just send us an email, and we will do our best in order to answer to the questions later than the webinar. And here's another question, however, this time for you, Cécile. Can you see it? Again, from Professor Cécile. How will the open IP question be resolved within the European Union? Sorry, forget this.
So the open IP questions to be resolved, do you mean, so you're talking about which ones, the ones connected to the tools, the ones connected to the allowability regarding the nature of the sequences?
Let's the participants be a little bit more specific. Gabi, please just write down what you're referring to, and I would go to the next question, question number 4, which is in the meanwhile, of course, we're going to come back to question number 3. Do we observe breeding companies moving out of the European Union because of regulatory difficulties? From Marian is this question.
Okay, so they're not really moving out, but they are moving their activities outside of the EU regarding this type of varieties. That's for sure. They develop edited varieties in areas of the world where the market is more open to them. And so they also, so it depends on their size, of course, but they can also start to get contracts with bigger companies to have access to some tools and to participate to the development of breeding initiatives. Far-away future.
Maybe
So,
I can add something about that.
Okay.
Yeah, see, this is the problem of the lack of harmonization and countries not really negotiating with each other the nature of their legislations. This is very clear in the study of the Commission. If you have the opportunity, please give it a look. And what is acknowledged by the Commission is that several countries outside of the European Union have different approaches, so you can categorize them in but they do not have the same approach. What happens when this is not the case and all countries are not really working together is that there is always a business opportunity for taking advantage of the difference in legislation. Now, this happens also on the litigation side with the forum shopping, where we try to choose the court that is more favorable to our interests. This should be avoided, and this is why the European Union should be more present on the international negotiation table to really make sure that we are on the same page with other countries. Thank you.
Okay. So to go back to IP question,
which was how will be the open IP question solved within the EU? To the essential, it was referring to the essential derived varieties.
Okay, so that's very precise now. Thank you. So this issue is already tackled by the UPOV office. That constituted a work group and that are hearing all the stakeholders. We have already, I think, 3 meetings where presentations were made by different breeders associations and by different personalities. So for now, the tendency, at least from the breeders, is to defend the fact that a revision of the concept should be done, and that it should be based more on the genetic similarity between varieties. So meaning that the genetic background will prevail over the number of mutations. And so that we're gonna stick to a situation where the initial breeder will be the owner of the family of EDVs. Not the owner, I'm saying anything, I'm sorry. That will be the one having to give consent for the commercialization of the EDVs. Do we think it will be possible in a faraway future that MBT could be classified as standard essential patents if they implement a market-dominating standard of technology, in which breeding techniques, I guess this could be the case? So Ursula, maybe I need your help. Is it classified as a standard essential patents? That seems like very legal terminology.
It is a legal terminology, but I'm afraid I don't have the technical knowledge to reply to that question. So maybe you can take our email address and we can try to help you, no? This is a situation where a certain patent is really essential to develop and use another technology like in 3G and 4G and stuff like that. So I think we would need a couple of minutes to think about some examples for that.
Yeah. Okay, so we're going to come back to, do you think that edited plant material is already imported into the EU market without being declared as edited? Am I authorized to answer this question? So from my very own personal point of view, I think there's some chance that it is already the case, because it's sometimes very, very difficult to separate the chains. But officially, no.
Yes, indeed. The result of the product is really undistinguishable. It's not if it's not declared. In many occasions, it's impossible to know whether a certain plant has an improved yield, for instance, just because it was crossed with the right pattern, with the right parents, or because of these new genomic techniques. And moreover, there is really a lot of complexities with the labeling of the products produced from these GMOs. So there is a possibility that there is a lack of declaration and of tracking and labeling in this field. So cannot be excluded. And this is why the adequacy of the legislation should not be only on the substance or on the definition of what's allowable and what's not, but also on the enforcement and the implementation of effective control measures on our territory, which goes, of course, together with the negotiation and being on the same page with other countries.
The following question is a little connected to the previous one, because it is indeed because we can't really detect, we can't control, that we cannot answer, and that we can not really stop importing edited varieties if they're not declared. So it's urgent to adapt the regulation. If we go back, Ursula, to question 5, so a market dominating standard of technology. I'm not sure I fully understand the question.
Yes, we can discuss this question later. Maybe we can get the contact of this person and we can try to answer to that later,
Okay.
with examples. Okay. So that we, the legal and the technical part of the CPVO talk to each other and come up with some examples.
Concerning the difficulties to identify STN1 and 2, how much time can you invest to adapt the corresponding rules in question?
This is a quite interesting question. Now, the EU, well, the European Commission has been preparing and working a lot already since 2011, like Cécile was mentioning before, in order to adapt the legislation, because the legislation in 2001 was quite specific, was the product, was a compromise text, was the product of a certain knowledge, and already 10 years after, it didn't age well, right? So it was already not really adequate. So as from 2022, there is already a program in the Commission of a revision of this regulation. So it's really working on it. However, if you follow the first part of the presentation of Cécile, she explained very clearly that this is a lengthy process, and in order to maintain the legislation relevant, you really need to hear a lot of stakeholders. Member states, associations, EFSA, the Food Security Agency also participated, for instance. So it's a very complex assessment. Also, you have to take into consideration ethical considerations, political considerations, and it's not easy to come up with an adequate text. However, the works to put something in paper are starting next year. I don't know if Cécile wants to add something on that.
You summarized it very well. That's exactly.
Okay.
So how much time can we afford to wait? So we have no time unless we consider that these products do not represent any risk, and then it's there's no need to rush, and we're going to end by declaring them out of the scope of the regulation. I don't know. No.
Okay, Michael, I don't know if that was the last question.
It was indeed the last question. There's just one comment from Marian, who's saying, congratulations on a very good presentation. I just wanted to let you know this. And so with that, I would thank very much once again our colleagues from the CPVO, Ursula and Cécile. It was a pleasure, like always, to collaborate with you, and most importantly, it was a pleasure to meet you today, Cécile. I hope we'll have the chance to collaborate again in the future. It was a very clear and interesting presentation, And also, I think participants have enjoyed that.
Thank you very much. It was a pleasure.
Thank you.
Thanks to you all, and I wish you a lovely lunch and afternoon. See you in the next couple of days again with the European IP Helpdesk, because we have a few webinars before the summer break. And if you'd like to join, you're of course very welcome to be with us. Goodbye.
Goodbye.
Bye.